So, hi everyone, my name is Jocelyn Martinez. I'm currently a first year PhD student at Texas A&M University, where I'm investigating the effects of short pulses of infrared light on metabolic coenzyme binding. Did you know that over 600 million individuals are living with neuropathic pain? This type of pain is caused by damage to our somatosensory system through either our central nervous system or our peripheral nervous system. Current treatment options to combat this pain include TENS therapy, pharmacological, and optogenetic methods. However, these methods are often not spatially precise or can cause further damage to an individual. So, therefore, an alternative method is needed to help combat this pain. So, a phenomenon termed as infrared neural stimulation has been shown to actually excite and inhibit action potentials in neurons if we introduce short pulses of infrared light to them. So, although we're not entirely sure of what's taking place, we do think that this method could be beneficial as it's label free, no dyes are needed, it's very non-invasive. The area of illumination is very spatially and temporally confined. And if we adjust our infrared light wavelengths, we can also achieve an adjustable stimulation depth to help target nerves deeper into our body. So some clinical applications that have been shown in literature of infrared neural stimulation include neural prostheses, optical cochlear implants, as well as central nervous system stimulation. So if we take a look at these graphs, we can see that overall, infrared neural stimulation is indeed spatially and temporally confined. And when comparing optical stimuluses to electrical stimuluses, we do not see any introduction of artifacts. So, although we're not entirely sure of the mechanism, we do know that short pulses of infrared light will cause a fast thermal gradient within our neurons. So essentially, photon energy will be converted into heat at that location. And at that location, we also see that the mitochondria will have a upregulation of calcium. And since we know that energy is needed for these cells to function, we believe that a tight regulation of metabolism is needed to help action potential propagation as well as communication between neurons. So this brings us to our project hypothesis where infrared light induced thermal gradients will alter neural activity through changes in enzyme kinetics and rates of cellular metabolism. So to help prove our hypothesis, our study will be performed with lactate dehydrogenase or LDH as our enzyme. So we picked this enzyme because it's very vital for organisms within the absence of oxygen. So essentially what happens during anaerobic glycolysis is that LDH will help regenerate NAD+ by transferring a hydrogen ion on NADH to a keto group of pyruvate. So overall, this binding between NADH and LDH will help pyruvate to be converted into lactate, and then hopefully cellular metabolism can continue when there's no oxygen. So an important factor to know is that our NADH exhibits something known as a fluorescence lifetime. So essentially, when you excite this NADH to a higher excited level, the time it takes to decay back to the ground state and emit a photon is called our fluorescence lifetime. We can then measure our fluorescence lifetime using a imaging method known as fluorescence lifetime imaging. Essentially, we want to be able to quantify our free or protein-bound fractions of NADH. So we take a look at our table, we can see that we can then calculate our lifetime using our equation. And then by fitting our intensity of our pixel images to a 2 exponential model, we will then be able to quantify our amount of free NADH, which is the amount of NADH that is not binding to our LDH in the presence of our IR pulses. So this brings us to our methodology. Here's a brief methods overview. So overall, this study will first derive a computational model. So to do this, we will import IR-induced thermal gradients, and then we can simulate different changes in the amounts of free NADH during exposure to a laser. We will then do a steady state temperature experiment. So essentially, we will use a stage top incubator to help mimic different thermal gradient temperatures. We will then capture fluorescence lifetime images. And as we previously stated, we would use those equations to then quantify our fraction amount of free NADH using a SPC image software. We will then do a dynamic temperature experiment model where we will then introduce our solutions to a 1470 nanometer laser with different pulse durations. We will capture fluorescence images and then quantify our fraction amount of free NADH using a custom MATLAB code. So here we see that the first step to make our computational model is to import previously published thermal gradients. These were induced by short pulses of infrared light using a 1869 nanometer wavelength at different irradiant exposures and different pulse durations. So as you can see on the graph, our results show that for a higher exposure, we would expect to see a higher rise in temperature of around 43 degrees Celsius compared to a 33 degrees Celsius for a lower irradiant exposure. We then were able to derive our rate constants for our NADH-LDH reaction using a Harnius equation of reaction rates. So this essentially is, the equation is K equals AE to the negative EA over RT. So we will then, based on literature, put our pre-exponential factors as 299, our activation energy for LDH as 10,000 joules per mole, and then we will put our thermal gradient temperatures derived into Kelvin as our T values. So here we can see that so here we can see that for a higher temperature based on our pulses, we get a higher rate constant. So then this means that our rate was being catalyzed at a faster rate. We then set up different binding parameters to help bind a 50 micromolar solution of LDH and NADH. So we wanted our NADH concentration to be 50 micromolar because it will diminish any self-quenching. We also wanted to set our alpha-1 values as 0.6. So based on literature, we found that our K dissociation rate constant should be 4.2 at room temperature. We then found, based on literature, that LDH will have 4 binding sites per enzyme. And then using these two equations on the bottom, we were then able to compute that our enzyme concentration should be around 13 micromolar. Also using the same two equations, which was previously shown, we were able to calculate our fraction amounts of unbound NADH. And here we can see that an introduction of a laser pulse will indeed increase our amount of free NADH, and then after the exposure, they will then decay exponentially. So next, we were going to experimentally verify our results. We conducted a steady state flame experiment. The purpose of this experiment was to collect different fluorescence lifetime images to see our changes in alpha-1 values. We used a custom made inverted multiphoton fluorescence microscope and a stage top incubator to keep our solutions at different temperatures based on our thermal gradient model. We then conducted data analysis using an SPC image software. And here we can see that for our steady state results, we saw different trends. We saw that a higher temperature will lead to lower lifetime. We also saw that a higher temperature will lead to more free bound NADH. So that means that NADH was not binding to our LDH as much. And overall, based on our percent errors from our model to our steady state results, we saw that overall they were not very high. However, at 43 degrees Celsius, we did see a higher percent error, which could be possibly due to model inaccuracy as well as contamination of our molecules, or simply our data analysis was not correct. However, based on these results, we believe that our model does match our steady state. So next, we were going to do the same experiment now, but with the introduction of our laser pulses. So here we can see that we did the exact same experimental setup, just now we did different pulse durations of our laser, and then we had to perform data analysis on a MATLAB code. So here is a representative image of our intensity values for a 1470 nanometer 0.5 millisecond duration pulse. And here we captured each pixel image as a 256 by 256. The time of our scanning was at the top left corner, and then it went from left to right and then top to bottom. And since we saw that our thermal gradients occur very fast in the order of micro to millisecond durations, we had to think of a method where we could image fast enough to capture all of our data. So we set our SPC dwell time as 100 microseconds, so each pixel was collected for 100 microseconds before being moved on to the next pixel and so forth. So when we tried to analyze our images on our SPC image software, we saw that we would bin in a cube. However, since we lose all spatial information of where our pulse is, we only are left with our temporal confinement of our illumination. So therefore, we had to think of a solution where we could bin in a line. So that's exactly what we did on our MATLAB code, where we binned by 8 pixels. So here are the results. When we binned all of our decay curves by 8 pixels, we saw that we were able to do this on our MATLAB code. So then we were able to perform data analysis. We continued with this and we then deconvoluted our data with our instrument response function. We found our maximum peak for each decay sum group, and then we fit a 2 exponential model to that decay curve to be able to quantify our free to bound ratio amounts. And overall, we do see that here we can see that based on our pulses, we do see the same trends that we would get at higher alpha 1 value with the temperatures, and then they would decay down after exposure. And overall, we do see that our free NADH does start around 0.6. However, this data does look very noisy, so we have to continue optimizing our data analysis methods to help get a better alpha 1 values as well as a better quantification of these changes. So in conclusion, we were able to experimentally verify our model results using a steady state temperature experiment, and the dynamic temperature experiment does in fact show the same trends. So we see that there is promise for this this model, and then we were also able to see that our infrared light does in fact increase our amount of free NADH, suggesting that cellular metabolism does change with the introduction of infrared light. So we hope that this study would potentially help us gain more insight into the mechanism behind this phenomenon, so we can then accelerate the future development of different IR technologies. And overall, we hope that infrared neural stimulation will be able to be used in the clinic as a non-invasive, non-genetic, and reversible method to help combat the neuropathic pain that we see today. So I would like to acknowledge all of my lab mates at the Quantitative Optical Imaging Lab, as well as our collaborators and our funding sources that help make these studies possible. So thank you so much. Hope you enjoyed this presentation.